a separate paper that is still in revision in Nature Immunology that shows that once these T cells are rejuvenated, and once these T cells are now capable of undergoing the telomere transfer reaction that I just described, these T cells then disseminate flow of telomeres in the bloodstream. They disseminate flow of telomeres in the bloodstream. These telomeres, they travel everywhere. It's a leftover of telomere transfer donation. They go everywhere. They go in the brain. They go in the gut. They go everywhere, and they rejuvenate each single senescent cell in your body. And this is responsible for extension of the lifespan in the animals that we are treating with the DOS compound. We're treating with this reverse of telomeres from 2.5 up to 5 years in the Black 6 mouse model, which is amazing. And yes, and we believe that this is the key, which is responsible for maintaining healthspan and lifespan. Meaning that if I'll give you the technology to rejuvenate your CD4 T cells, which are responsible for this process, I can extend your lifespan. I can improve your healthspan.
Hello, Dr. Lanna. You are the founder and CEO of Senscell, a company developing human rejuvenation therapies with rational design. So welcome to Modern Healthspan, and thank you so much for joining us today.
Thank you for the invitation. Yes, I'm the founder and CEO of Senscell. Senscell, we focus on this, I would say, unanswered medical question, which is how we modulate the immune senescence of T cells. If you think about other pathways, such as regulatory T cells or exhaustion, immune senescence has been largely overlooked, meaning that to date, there is not a drug that can actually target these immune senescent cells to rejuvenate them. So immune senescence can be defined as all sort of bad things that happen to immune cells when they grow older. And this causes a bunch of problems, starting from a lack of responses to vaccination, the inability to control infections, the surge of cancers with age, and also nowadays this has been connected with neurodegenerative disorders, for instance. However, we're moving forward to the awareness that if you regulate the youth of a single cell type, which is the CD4 T cell, you can control the overall lifespan and healthspan of the organism. And this is what our data show. In order to do this, we've been focusing on a complex of proteins that are characterized during my university years before founding Senscell, when I was a PhD student at University College London, which is called the SMAC. The SMAC is a complex of stress proteins bound together that dictate aging phenotypes to the T cells. So, by disrupting the SMAC with the first-in-class compound, we can impose a rejuvenation on these cells, and we can change their aging fate. We can reprogram them to give them new functions.
Okay, so one thing you said a CD4 positive T cell.
Yes.
So
yes.
What is that? I mean,
so yeah. Well, if you have heard of HIV or what it comes with the infection, you probably know that when these T cells are attacked by a virus, you essentially, if it's uncontrolled, people die, right? So CD4s are the lead cell that governs all the immune response. They are essential for instance, the response to vaccination, and they coordinate the entire immune response. So they are the pinnacle of the adaptive immune response. So if you can rejuvenate these T cells, so if you can control their health, you can control the entire process.
Okay, so as the immune system gets worse with age, as we get this immunosenescence, it has a larger impact than just the immune response. Is that correct? I mean, do we
Yes,
see
correct.
Other negative?
Yes. So that's exactly what I was trying to say. So the new data. So we started like 10 years ago, understanding that immune senescence is bad for things like they're restricted. Initially, we thought they were restricted to the immune response, and now we are actually moving towards a new redefinition of the process, such that when bad things happen to the immune cells, bad things happen to the entire body, right? So that's why it's super important to have target new therapeutics that can rejuvenate these cells, that can take care of these cells. Doing so, you take care of the entire body. So yes, you're correct in saying immunosenescence, the deterioration of the immune response, also has an impact on non-immunological functions.
Right. So we see that rejuvenating the immune system will effectively rejuvenate the body.
Yes.
Yeah. So that okay. And so now we have these T cells, the CD4 positive T cells, which are the ones that are active, and they're the non-naive ones, right? They're the ones that are trained to attack specific antigens. Okay.
Yes.
So how do they get old? What happens to them as they age and they become senescent? Is that correct?
Well, yes. Essentially, we have recently somehow redefined this question, because the dogma in the field for 20 or 30 years has been that when a naive T cell, so meaning a cell that has never seen an antigen before, sees an antigen, then become an effector cell, then a memory cell, and after repeated encounter with that antigen will be driven to senescence, right? So that is the model, saying that, every round of division, the ability of the T cells to upregulate an enzyme called telomerase, which is quite famous and takes care of the end of the chromosomes that are known as telomeres. They get shorter and shorter with age. So because of their inability after subsequent stimulation of reactivating the telomerase, the T cell then will eventually become senescent. But a couple of years ago, we have shown that actually this is a bit different, because actually the aging decision of the T cell is defined before the naive T cells undergo the first cell division. Because when they encounter an antigen presenting cell during an immune response, there is a decision whether that specific T cells will receive or not a telomere donation from the APC. These telomeres travel across the synapse, so the antigen presenting cells behaves like a telomere donor cell for the T cells, which is kind of surprising. And once they get the telomeres packaged in vesicles, they incorporate this in the telomere of the T cells, of the receptor T cells. They extend very much, 20, 30 times more than what a telomerase can do. And that T cell that has received telomeres before the first decision will become a stem-like T cell, so will not become senescent. Conversely, if they do not receive a telomere donation, that T cell will become senescent in the future. So it's totally unrelated to the telomerase. The telomerase comes later on and actually support the expansion of the effector T cells. Is important to support to fight the infection. So when the T cells has the information, receive the information from the DC, from the APCs to fight the infection, that T cell will mount a very large immune response. So they will proliferate greatly. And that telomere is important to drive that proliferation, but it's nothing to do with the aging phase decision of the T cells, which is established before the T cells has even divided once.
Yes,
I published by myself in Nature Cell Biology 2 years ago.
Right. Yeah. Yes, I saw that, and that mechanism where. So what was the APC? Sorry,
So the APC is an antigen presenting cell. Could be a dendritic cell. Could be a macrophage.
Okay, so the APC donates telomeres like in a vesicle, and then the T cell like attaches them to the end of its chromosomes.
Yes. Yes.
That's amazing.
It's amazing, but to think about it, think about the role of telomerase. What a telomerase can do, they synthesize 100 to 200 base pairs of telomeric material, which is serving as a buffering mechanism, because every time a T cell divides, roughly that amount of telomeric DNA is lost. So essentially what the telomerase does is, okay, the T cells will lose 100 to 200 base pairs of telomeric material. What the telomerase does, it puts back that sort of material. Because of this, we always thought, we've been thinking this for 40 years. Telomerase got the Nobel Prize in 2009, and everyone thinks that is the key to rejuvenation. However, nowadays, telomerase has not been exploited successfully to achieve rejuvenation. So probably it's not the full picture, right? So the fundamental problem and caveats with this is that it's not taking care of the ultra short telomeres. So telomeres that are ultra short are those that are defined below a certain threshold, let's say 2,500 base pairs, right? So 2.5 kb. So the telomeres cannot even attach to these telomeres, and even putting back 100 base pairs in each cycle will still qualify these ultra short telomeres as ultra short. And these are essential because the ultra short telomeres trigger senescence. So what the telomerase does by pushing the T cells to divide, and when these ultra short telomeres accumulate, it's actually signaling senescence signal to the cell and driving them to senescence. So it's the other way around than what we thought. Instead, the ultra short telomeres are completely eliminated by telomere transfer. So when you get a telomere transfer donation, so the telomeres, because they extend so much more, the ultra short telomeres, which are not elongated at all by telomerase, it feeds into these ultra short telomeres. They are extended before the first division by 2,000 base pairs, and these telomeres will not be ultra short before the cell divide. So the information of senescence-related pathway will not be starting if there is a telomere transfer donation. Conversely, if the telomere transfer does not occur, that naive T cell will still have ultra short telomeres. Yes, also the naive T cells can have ultra short telomeres. It seems weird, but it is true. And very few, 1, 2, or 3, but even one ultra short telomere can be enough in the future to dictate a senescence fate. Of course, I'm not saying that it will become senescent in one round of division. What I'm saying is that if we do not take care of the ultra short telomeres, at the very early stage in the life of the T cells, that T cell will become senescent in the future. The only way for the T cells that we know now to take care of the ultra short telomeres is to receive a telomere donation from the HPC. If they receive a telomere donation, the ultra short telomeres are eliminated because they are extended, right? They're extended by 2,000, 3,000 base pairs before the T cells engage in mitosis, before the T cells engage in mitosis, but before the telomere is even active. Before the T cell engage in mitosis, the ultra short telomeres are elongated, and therefore that T cells will now divide without an ultra short telomere burden. That's what we call.
Just out of interest, do we see this mechanism in any other type of cell, or is it only T cells?
Everywhere.
Everywhere.
Even in plants.
Okay. Yeah.
Well, that's what we have discovered. We haven't published it as yet, but I can anticipate it's everywhere. It's in the skin. It's everywhere.
Okay, we'll leave that because that would be a total diversion. Okay, so we're stopping this T cells from becoming senescent. We're giving them new telomeres and we're rejuvenating them. So
we're preventing we say that we are, by giving the telomeres, we are preventing their aging fate, because we define rejuvenation as intervening on the T cells that are already senescent. So by telomere transfer, you make sure the T cells do not become senescent in the future. If you want to rejuvenate T cells that are already senescent, you have to do something different. You have to destroy this complex of proteins I was talking about before.
Right, and we will get there. Okay, so this telomeres lengthening process is going on anyway. So does your intervention cause this to go on more, or your intervention
it restores it, because the capacity to telomere transfer is lost by the T cells, which is senescent. So when I rejuvenate the T cells now is able to do again once more the telomere transfer mechanism.
Okay. So why does removing the senescent T cells kind of help with inflammaging? I mean, so it reduces inflammation. Is that correct?
Yeah. Well, we are not removing technically, because this is done by people who are trying to, approach this with senolytics and killing the cells. What we do, which I think personally, is a very dangerous proposition when it comes to the immune response, because you think about someone who is let's say about 65, you have thymic involution and so on. So, and you can have like roughly 50% of the immune repertoire actually made, or even more, made by senescent cells. Now, it doesn't take a genius to think, if I kill 50% of your cell and you don't have the ability to make new one, you might suffer from immune deficiency, right? So what we instead do is completely different. So we see these senescent T cells as a resource. So we leverage on them by rejuvenating them. So we don't kill the cell, we reprogram it. It's a sort of, I would say nowadays it's fashionable, somehow a green light technology, so I like to call it, because it's recycling the cells that are thought for you throughout their life, and then now they are senescent. Instead of getting rid of the cells, we reprogram the cells, we turn the biological clocks back in time, and we now discover, we give them to new function. But this is related to the TCR. It's not answering your question. To answer your question instead, we have a separate paper that is still in revision in Nature Immunology, that shows that once these T cells are rejuvenated, and once these T cells are now capable of undergoing the telomere transfer reaction that I just described, these T cells then disseminate flow of telomeres in the bloodstream. They disseminate flow of telomeres in the bloodstream. These telomeres, they go everywhere, they travel everywhere. It's a leftover of telomere transfer donation. They go everywhere. They go in the brain, they go in the gut, they go everywhere, and they rejuvenate each single senescent cell in your body. And this is responsible for extension of the lifespan in the animals that we're treating with the DOS compound. We're treating with these reverse of telomeres from 2.5 up to 5 years in the Black 6 mouse model, which is amazing. And yes, and we believe, we believe that this is the key, which is responsible for maintaining healthspan and lifespan, meaning that if I'll give you the technology to rejuvenate your CD4 T cells, which are responsible for this process, I can extend your lifespan, I can improve your healthspan. And 5 years, in terms of corresponding human life, we're talking about roughly 120 years.
Yes. Is that published yet, that mouse?
Not yet.
Not yet. Okay. So
since you've asked.
But I did not. Yeah, that piece I didn't gather from my research.
This is still not on the public records, but
right.
You asked, so I feel to share.
Thank you. Yeah, that was good. Okay, so I did see one thing was that rejuvenating, unsenescing the T cells may help with thymus regrowth because it involutes, right, and it turns into fat. Have you seen that, and do you know what the process is?
Yes. So when you do this process, also the thymus grows back, and the T cells. This is so much related to the rejuvenation process, because this is the paper now we have under revision in Nature Cell Biology, and this is so much related to it and this is the paper also which is I can talk more freely because on the public records as a preprint. What was very surprising to us, to me, is that always what we've been thinking is that when you rejuvenate something, what is rejuvenation? What is rejuvenation? We thought that rejuvenation would only be restoring function lost with age, right? So, an ability of the cell that is lost with the differentiation or with age, then you rejuvenate the cells and you gain back the same ability. And actually, we have learned from our studies on the T cells is that this is not the case, because when they are rejuvenated, they will still do that sort of task, but they learn new functions because they reprogram their TCR beta gene. They reprogram TCR are these proteins that are on the cell surface of the T cells that are essential because they essentially connect the T cells to the foreign environment, and they recognize antigen which was presented by the antigen presenting cell, right? So they are on the surface. What the rejuvenation does to these T cells is that it reprograms the TCR. So it changes the specificity of the TCR. So you can tell the T cell, I want you to become now a cancer-specific T cell. I want you to become now whatever you think of. Yes, so the new TCR, essentially you can train, so the new TCR will respond to pretty much anything you want it to respond. So for instance, what we have to prove principle of this, we use as a model yellow fever vaccination, because in the West, people are naive to yellow fever because they don't get vaccinated to yellow fever. We can generate overnight, starting from fully differentiated senescent T cells without the vaccine, with this rejuvenating compound we have developed in the presence of yellow fever peptides or anything you want to do it, and that T cells will become yellow fever specific. So in order to do this, because of course some TCRs, the mechanism is not 100% generating specific TCRs, so obviously potentially autoreactive TCRs are generated, but remarkably, there is no sign of autoimmunity whatsoever in the animals which are living up to 5 years. And the reason is that these T cells that are reprogrammed, we found that they go back to the thymus in the animal and in the old animal, and they once more they get their TCR in check. Even if they are mature, fully mature, these are fully mature T cells that are senescent, they've been rejuvenated, and they migrate back from the periphery to the thymus. In the thymus, they undergo once more thymic checks. They are called negative selection processes. And these processes eliminate roughly, we found they eliminate roughly 30 to 50%, 30% roughly of the revised TCR, meaning that the remaining 70% leaves the thymus once more, and they are fully tolerogenic T cells that can fight infections. And these are moving us towards new awareness such that is getting vaccination redundant, because what we have compared it vis-a-vis with vaccination. You can, but we started from the problem that told people do not respond well to vaccination. We wanted to improve it, and actually then we ended up understanding that perhaps we found something that works better than vaccination. You can perfect the vaccination process, but you have to choose between vaccination and the rejuvenation compound. The rejuvenation compound, at least in the preclinical data we have, is so much more powerful and effective at protecting animals from lethal infections than the clinical approved influenza vaccines, for instance. And the way it works is by reshaping these immune T-cell repertoires with these specific TCRs that are tolerogenic, and even without vaccination, you could receive the rejuvenating compound, and you will shape, your T-cells will shape their TCR depending on the future challenges they will receive. So the fundamental difference is that, whereas with vaccination, you have to program somehow the sort of response you want to mount. So if you want to be protected for influenza, you will receive an influenza vaccination, right? If you want to be protected for COVID, you will receive a COVID vaccination, correct? Instead, what I'm talking about, and of course, it doesn't work very well in all people. What I'm talking about is that if you receive a rejuvenating compound and you now reprogram this army of soldiers with reprogrammed TCRs, you don't have to predict the challenge that you will receive, because those T cells, they have the flexibility to reshape their T-cell repertoire according to the challenge they will cope with in the future. And we've done this in the animals. So they've received a rejuvenating compound injection, and 6 months, even 6 months later, or 3 months later, they get an unrelated infections, and these animals are fully protected from the infection, even in the absence of vaccination. And we're talking about old animals.
This ability to reprogram the TCRs, is that available in very young animals? Or, I mean, is this something totally new?
Well, this is something that is a good question. This is something that I think people, prominent scientists, somehow overlooked. There was a lot of, science, and sometimes is fashionable. So if you look at, of the production, there is intense research in an area, and then somehow scientists get bored and move on. So if you look at research that has been done like 25 years ago, in the early 2000s, year 2000, so there was a lot of studies on a process which is called TCR revision, which is exactly this process I'm talking to you about. But people did not understand it. They thought that this process was a process that would allow the T cells to comply with their defective expression of the TCR to make it a new one. Actually, nothing was more wrong, because those people, they were looking at those T cells, those stem-like T cells that were revising their TCR in order to expand the immune repertoire. So there's nothing defective about the process, or the reason why the process is in place. Actually, this process is done by the healthier T cells that want to expand the TCR repertoire. It happens at a very low scale, because the stem-like T cell compartment in man is about 1%, roughly 1%. Obviously, with the pharmacologically, we can boost the process, and we can boost the process to 20% of the repertoire, 30% of the repertoire, and thereby now we are able to observe it, we're able to study it, we're able to manipulate it pharmacologically, and to exploit it as well therapeutically.
You have developed this intervention which rejuvenates the T cells. So the first thing is, you said that you're using rational design. Yes. Could you talk a little bit about that? I mean, how do you use rational design to come up with something to do this rejuvenation?
Yeah. Well, it's a very simple process, because rationally I knew So the process is based on this complex of proteins I have characterized over the last 15 years, which is composed by many proteins, but the proteins of interest that we are targeting are called senescents. Senescents are a family of stress-related genes that are expressed by the senescent T-cells, and they are bound to another important protein, which is called AMPK. AMPK is a crucial metabolic regulator in the cell, is activated during starvation or is activating upon DNA damage as well. And senescents, during these stressing processes, like starvation, DNA damage, and so on, they bind to AMPK and activate AMPK. So what the rational design is based on the fact that I knew from, I would say, truncation studies, the domain of senescents responsible for the binding to AMPK and to activate the entire complex of proteins I described in senescent cells. So simply what we have done, very, very simply, we got the lead in 2 weeks. Very simply, we did segmentation of these 160 amino acid domain of the senescent proteins. We've done peptide scanning and found that the peptide which was responsible to interfere with this sort of a binding. And then we made a cyclic peptide out of it, and we called it a DOS, a disruptor of the SMAC. The SMAC is a complex of protein made of senescents, AMPK, and MAP kinases, which I've described in the senescent cells over the years.
Okay. And so that is the intervention, which is this cyclic.
And rational design is because we didn't do, I wouldn't say we didn't do blind screening. So we knew which part of the proteins to modulate and to attack.
Right. Okay. And so, yeah, so it breaks up these complexes, and then that allows the T cell to perform its function correctly, because somehow these complexes form as the T cell gets senescent.
Yeah, upon DNA damage. The senescents are transcribed by a very famous protein, which is called P53. P53, as you might know, is the king of senescence and apoptosis and all these bad things. So when there are stressors, P53 gets senescents to be transcribed. So the cells express senescents, and these senescents then will form these SMACs in the old T cells. What we did not expect, though, because obviously, if you think about the cyclic peptide, they have a very short, lifespan, I would say, half-life in vivo, because they're quickly degraded. So we thought they would be able only to block this SMAC signaling cascade for a very short amount of time. And instead, surprisingly, we discovered that in animals treated with a single injection of the peptide, which is eliminated in less than 2 hours from our PK studies, actually they persist in a state of youth which is almost permanent. And even 6 months, 1 year later, these SMACs are not there, even if the compound has only been given 1 year earlier. The reason is that we identified a full signaling cascade whereby the disruption. So essentially, the good news about the aging process is that it reinforces itself through feed positive cascade. I like it to call it feed positive cascade, meaning that, think about the P53 example I told you. There is a stress, P53 is activated, senescents are transcribed, senescents form these SMACs, SMAC activates P53, and P53 transcribes more senescents. So if you break into this process, you can destroy the entire process, not only the expression of the SMACs, but also further transcription of the proteins, and also there is epigenetic silencing of the senescent gene. So meaning that a single injection, you get rid once of the protein, at the protein level of the complex, and you also make sure that in the future, there won't be any more protein complex to be formed. So we have a permanent, strong reduction of the SMACs in this. There is, of course, a trace of the SMAC in the T cells that are rejuvenated. I'm not saying that these are limited, and also we don't want to do that for safety reasons. I'm not saying that these SMACs are eliminated by 100%. I'm saying that we get roughly an elimination. I would say, well, it's donor dependent, can range between 80 to 30% of the elimination, but that is enough to reprogram the cells to stem state. So they will remember they were all cells before, because they will have some trace of SMACs as compared. So they are completely identical to a T cell that is young, but they have some trace of SMACs, but in function, they have this expanded function, the expanded repertoire, because they have new TCRs and they can protect you from challenges.
So these SMACs, do they only form in T cells? Do they form in other cell types?
They form everywhere, and if you look at beta-galactosidase, for instance, which is a famous senescence marker, every cell which is beta-galactosidase positive also has SMACs.
So would potentially reverse.
Interchangeable. It can change. You can rejuvenate the entire body, but also, as I mentioned earlier in the call, simply by rejuvenating CD4 T cells, because the T cells then has this function, the stem T cells, they sit in the lymph nodes and so on, and it's like Spider-Man. They release all these beautiful rivers of telomeres in the bloodstream. They go everywhere. So even if you do not target the SMACs directly with the drug, simply by rejuvenating CD4 T cells, the T cells they can then donate these telomeres. We know this because simply what we have done is that we took T cells that are rejuvenated with this disruptor of the SMAC. We transplanted these T cells into old animals that did not receive the disruptor of the SMAC compound, and the rejuvenated T cells can rejuvenate the animals. So even the rejuvenation process is transferable. I can transfer rejuvenation from an animal to another, from a being to another being, not by injecting the compound, but simply by offering them the rejuvenated T cells, or offering them the reverse these rejuvenated T cells they produce. So these stem T cells are beautiful. They are sitting in our lymph nodes, and in the resting state, what they do, so you don't have to engage. Or I mentioned all the beautiful things about the TCRs expanded. That happens if there is a challenge. If there is not a challenge, they are beautifully sitting in the lymph nodes while they are resting. They are slow pace releasing these flows of telomeres from the lymph nodes. We took these beautiful pictures of all these rivers that connect the efferent vessels with the circulation, and all this flow of rivers, they go everywhere, and they take care of all the beta-galactosidase positives in the body. So they rejuvenate the entire body. So the animals, they change in appearance. The animals, and I'm not talking about, for instance, David Sinclair epigenetic modeling, which is a genetic model. I'm talking about a natural rejuvenation process, which is entirely governed physiologically by the reprogrammed stem-like T cells.
Interesting. So you've kind of referred to the trials that you're running, the preclinical trials that you've been running. Is there anything else from the preclinical trials that you would like to share in terms of what you've seen for rejuvenation of animals?
Well, what clinically, talking about trials, so clinically, the most exciting thing that we have seen, so because we want to perfect, we want to enter perfecting the vaccination process, because, if you think about vaccinations, and you think about the addition of adjuvants in the modern vaccine formulations, or even people have been trying with high doses vaccines, all these are things that somehow been quite disappointing, I would say, because in the improvement over non-adjuvanted formulation has been consistent but very modest, and there's still a big problems in terms of immune protection. So the most exciting thing of immediate clinical value, and this will be the basis of our clinical trial starting at University College London in 2025, will be to add the rejuvenating compound to the clinical vaccine and show, because at least this is trying to show, because this is what our preclinical studies have shown, and show that you can restore the immune response now and fill in the gap that exists between a young immune response and an old immune response with an adjuvanted vaccine. For instance, in animals, you offer them, you compare old animals with young animals once you have offered them a clinically approved influenza vaccine, and you can still detect there is at least 50% still of reduction in terms of immune response. And also you can measure that quite easily with antibodies specific to the flu challenge, for instance, upon flu vaccination, or even in terms in animals at least of immune protection from a little challenge. What we found is that you have at least 50% of reduction in old animals compared to young animals when they are vaccinated with a vaccine, with influenza vaccine, which is found in literature. What we found is that if you infect them with a little challenge, even if you fully vaccinate the animals, and old animals, you still have roughly 20% of survival only, compared to 100% of survival of a young cohort which is vaccinated. When you give the DOS with the vaccine, the survival of the old animals that before they were surviving 20% is fully restored at 100%. So you have a full restoration of immune protection. And the amazing thing is that if I now take out the vaccine from the picture and I only give the rejuvenating compound, the protection of the animals is 85 or 90%. So meaning that the delta between the rejuvenating compound and the rejuvenating compound plus the vaccine is not more than 20%. To say the least, at best. So all the immune protection which we are getting from, and I think we should talk about this a lot, because all the protection we are getting from the clinically approved vaccine is, quite reduced at old age. At least this is what our clinical studies are showing.
Right. So the human trial is starting next year.
Next year, yes.
And do you have any idea on how long is it going to take? And
the phase 1, which we expect to be very smooth, because another amazing thing about these compounds is that in terms of the NOEL, the NOEL overlaps with the maximum feasible dose, meaning that we observe no adverse effects up to the maximum feasible dose we could administer to the animals, which is over 200 times or 300 times the clinically approved dose. So we have the preclinical dose at which we see the effect of interest. We're using the compound at a concentration of 0.05 mg kilo. Think about other therapeutic compounds that have been used at 20 mg kilo, 30 mg kilo. So very short amount of the compound, very large therapeutic window. So the phase 1 for safety shall be very smooth. So we expect it to be ended in less than 1 year and then get into phase 2.
Interesting. And so in terms of approval, what you're looking to do is to show that you get better response from vaccines.
Well, it's a complex topic, I would say, because so we will start the testing with vaccination, to perfect vaccination. We will also have a group of people that do not get vaccines but only get the rejuvenating compound, to see whether they can be protected in the future from unrelated challenges or from infections and so on. Obviously, we need something to measure. So in order to measure something, the easier thing is to measure the response to the vaccine. So we will be relying on the vaccine response, but we will also, on a side, we will monitor the ability of people who are on the rejuvenating compound, which is called DOS, who are on DOS, to cope with unrelated challenges with or without vaccination. So we do want to have one arm of the trial without the vaccine, just with the DOS, just to see how those people, for instance, how many infections we report during the winter, not only for infections, any sort of infections, and they can be, of course, they can be not only reported, but can be documented in the laboratory. So we want to see, because we think that by rejuvenating these T-cells with the DOS compound, these people will be better off, for instance, against seasonal flu and so on, even cancers. But of course, we have a short amount of time. So we have only the observation. Of course, the observation will be short, like 1 year or 8 months or 9 months, but it's still clinically relevant. But obviously, the decisions will be initially based on the response to the flu vaccination. But we are very much aware that this is just the beginning and so much more can be done.
Yes. Yeah, I was just thinking, from my understanding of how the approval process works, is it needs, just people being less sick would not.
Yeah, of course. Exactly. So, well, we will have, as I mentioned, the primary endpoint will be the response to the flu vaccine.
Right.
But we are still getting, other collateral that are of interest.
Right. And it's a peptide. So hopefully the regulatory path is reasonably well trod and understood.
It's a small molecule.
Yeah. Okay, because some of the other rejuvenation, like partial reprogramming or whatever, I see as being far more complex in terms of getting them through approval because they're gene
From
technologies.
our interactions with regulators, because the molecule is very small, it's only 5 amino acids and it's below 500 Dalton. So this has been looked at as a small molecule.
Right. And this is in the UK. You're
Yes.
going, yeah, okay. Okay, excellent. Now, I looked at your website, and if you're free, can you talk a little bit about how this could be widened to other uses? Because I saw that your pipeline has like some anti-cancer and anti-neurodegenerative um, uh, molecules. So yeah, can you talk about how this technology might be used for wider uses?
Well, you see, what we have done is that we have engaged in exercises, applying, this sort of concepts to the asthmax, to all the biogenesis of the asthmax. So we have other molecules, like, for instance, which are called negators of the asthmax, which are molecules that negate the genesis of an asthmax. So they can intervene by preventing this formation in young cell. Or other molecule, which we call the mobilizers of the asthmax, that activates an asthmax, and we've shown that in some circumstances you can activate the asthmax, and that provides a very strong effector response, which can clear resistance infections, and it could be of use in sepsis, for instance, and so on. But because of all this is very exciting, but we somehow decided to, for now, focus on the disruptor. And this has moved from this new amazing discovery, which we have done, in which we actually found that the disruptor is offering a cure for HIV infection. Yes. So it targets the HIV, which nests into the DNA of the T cell. I cannot tell you how as yet. I know it, but I can't tell you as yet. Because you need to protect it, but it eliminates the provirus in the T cell completely in 4 hours.
Wow,
yeah.
that would be big.
Yes. So we've done this in, of course, for now we've done this in vivo studies. We've done this in vitro human studies on CD4 T cells, which are infected by the virus. The virus just disappears when the T cells is offered the DOS. So we are moving towards clinical trial to test this, and we're excited about this because you think about people living with HIV and the burden that the lifelong regimen to which you have to comply in order to control the infection. And we think that we can strongly reduce that burden, and we think that HIV can be cured with a few DOS injections.
Okay, that would be wonderful. Yeah. And it sounds like it would be really nice, pretty good just to have as I guess, as a rejuvenating thing, just to kind of help the immune system as we get older. So when you're 50 or 60.
Mm-hmm. Mm-hmm. Well, now we are seeing it. Unfortunately, I have to say we are seeing it even that investigating blood from people who are 30 and above and seeing that they still benefit from the DOS. Perhaps an effect of COVID, we don't know, but we are seeing that the platea of people that could benefit from rejuvenation is not anymore restricted to 60 or 50 above, but actually people who are 30. Yesterday, I was seeing people who were 31 having 40% of T cells that are senescent, and they were so much reactive to the DOS. So we also need to somehow reimagine, the sort of platform and people who actually may benefit from the intervention. Perhaps, obviously, to start, we will start from people in the trial who are 45 or 50 plus, but we are very much aware that even people who are much younger, they could still benefit from the technology. And I think this is what also regulators want us to do.
That is amazing. So if people wish to see get to find further information, where should they go?
They can contact me through the contact form on our website. They can find any of my papers when I'm a corresponding author and click on my email address. They can find me on LinkedIn, so they can find easy access to me. We are about to raise Series B capital, so if people are also investors are interested to reach out, they can.
Okay, excellent. And that is SenseCell. We will put the link into the description.
Sure.
Okay, Dr. Lanna, thank you so much.
Much of a privilege. I enjoyed it so much. Thank you so much for the invitation.
Yeah. No, thank you.